Hello, welcome back to the second part of the video series. Okay, till now we have been discussing about the transcription process. First part was regarding how the transcription took place in prokaryotes, then how transcription took place in eukaryote. While teaching eukaryote, I have already mentioned that the eukaryotic process of transcription was mainly of three processes. First was the regulation part. Second was the actual transcription process part, that is initiation, elongation and termination. And the third and the final part is the post-transcriptional modification. You must be wondering why the transcriptional process need to be modified. Because after transcription, the mRNA which is formed after the transcription is really very long. Okay, and there are certain parts in the mRNA which are not at all needed for the protein synthesis, or they do not contribute in the process of protein synthesis, like the intron. Okay, that's why the mRNA needs to be matured. The mRNA which is formed by the process of transcription is pre-mRNA. Why we are mentioning the term pre? Because they are not matured, that is they are not able to be, they are not mature enough to form the protein. That's why to form the mature protein, we need to make the mRNA ready so that it will form the transcription, so that the translation process can take place smoothly. For that, there is certain modifications is to be needed for the processing. Okay, the first one is the capping, second is polyadenylation, third is splicing process. In this part of the video, we will be only concerning about the capping and the polyadenylation process, and in the next part we will be discussing about the splicing process. Okay, when I was teaching you about the mRNA or the RNA structure, the mRNA structure, you must be remembering it, that the mRNA contain, the mature mRNA contain 5' and cap and 3' and tail region. Why? Why the cap region was essential? Because for the proper binding of the ribosome. Okay, that's why the capping process is essential. And what are the functions of all these two? We will be discussing each one in detail. So let's begin with the process of capping. So the process of eukaryotic RNA polymerase 2, as we have discussed that the RNA polymerase which take part in the mRNA formation is RNA polymerase 2. If you remember, RNA polymerase 1 was responsible for the formation of rRNA, 2 was for mRNA and 3 was for tRNA. So that is RNA polymerase 2, there are certain process which takes place, that is capping, splicing and the cleavage and polyadenylation. Okay, so let's begin first with the capping process. What is the capping? The tail of the before that, let's understand that the RNA polymerase, in the previous class also I have told you that the RNA polymerase contain a heptad structure in the tail region known as the CTD region. Okay, this is the CTD region, which is heptad region, and this region is responsible for recruiting the capping process. How it performs? Let's see. The tail of the RNA polymerase 2 contain 52 tandem repeat of T amino acid sequence. Okay, 2 serine in each repeat. You can see there is 2 serine at each repeat. Okay, next, capping protein first bind to the RNA polymerase tail. Okay, if you can see that this is the capping protein, this blue color, if you can see that first the capping protein will bind to that of the CTD region, that is the tail region, you can see. Okay, bind to the polymerase tail when it is phosphorylated on the serine of the heptad repeat late in the process of transcription initiation. So after the first, what happens, this capping protein first bind to that of the CTD region. After the CTD region is phosphorylated at the fifth position, fifth serine position, you can see phosphate is written. After the fifth position it is phosphorylated, this capping protein is added and the initiation is later on, the transcription initiation is begin. RNA polymerase is capped as soon as the 5' end emerges from the RNA polymerase. So you can see here, this is the RNA polymerase, and you can see here that this is the 5' end of the mRNA, that is it is still not yet formed. Okay, Before that only the capping protein binds to the RNA polymerase. Remember that. The RNA polymerase is responsible for formation of protein, mRNA. Before that, this tail region, in this tail region only, the capping protein binds to it, and the phosphorylation takes place at the 5th position of the serine residue. Once this 5 prime end will be emerging out, you can see this is the 5 prime end will be emerging out, immediately this cap will be attached. So this is the cap. Immediately when the 5 prime end will be emerging out, the cap will be attached to the 5 prime end. Okay. So the capping is the first modification. This is very, very important. Mainly it is asked in exam that which modification. There are 3 modification: capping, splicing and polyadenylation. So which process takes place first when the mRNA is formed? Remember, first the capping process takes place. Immediately when the 5 prime end comes out, the capping is done. Okay. And transcribed RNA takes place co-transcriptionally in the nucleus. This is very, very important point. What do you mean by co-transcriptionally? That is, along with the transcription process, the capping is done, not after transcription. Along with the transcription, the capping is done. This is very, very important process. How the capping is done? Let's see. So this is the mRNA structure. If you see, this is the mRNA. This is the 5 prime end of the triphosphate group is present. Okay, at the end terminal, this is the end terminal, and there are 3 phosphate are present. That's why they are triphosphate. This one is alpha, beta and gamma. One of the phosphate will be released out, as a result of which 2 phosphate will be remain. Okay. Next, what will happen? Guanyl transferase enzyme will be added, as a result of which the guanyl transferase also has 3, that is GTP. Okay, GTP has 3 phosphate group, that is alpha, beta and gamma. Okay, what happens? The 2 of the phosphate group will be removed. Okay, only the 2 of the, that is the beta and the gamma. Okay, the beta and the gamma of the phosphate group of the GTP will be removed in the formation of PPI. Why it is called PPI? Because 2 phosphate is removed. Here only single phosphate, that is, remember that which phosphate is removed? Gamma phosphate. It will, don't create confusion among that alpha. No, gamma phosphate is removed in the first case. Okay, first case the gamma phosphate will be removed. Here this is alpha and this is beta remaining. Okay, remember that. Then what happens? The guanyl transferase enzyme will come. Guanyl transferase, what is the role of guanyl transferase? It will transfer GTP. Okay, GTP means guanyl triphosphate. Okay, so it has also 3 phosphate group. So out of the 3 phosphate group, the beta and the gamma phosphate will be removed by the formation of phosphate PPI. So only the alpha position will be remaining. Then what will happen? This alpha position will be attached to it. You can see that is GP alpha and P beta and P alpha. This is alpha and beta of the mRNA, and this is of the GTP. Okay. Next, what will happen? Guanine 7 methyl transferase, that is to the 7th position of the mRNA group, what will happen? A methyl group will be attached. Which is responsible for it? That is adomate. Adomate means S-adenosyl methionine. It will methyl donor, or it is known, it is an enzyme which transfer the methyl. Okay. So the first methyl transfer is known as the C0. Cap is known as the C0. Then again the same adomate will be added. Again there will be methyl transferase at the 2 prime position. Remember that. Wait 1 minute. Okay. Wait first methyl transferase will be at the 7th position. Okay. This is the, that's why M7 will be written. Second one, methyl transferase, will be at the 2nd position. Okay. So 2 methyl group is attached, and the second capping is known as the cap 1. And the bond which is found between them is 5 prime to 5 prime triphosphate bridge. This is very, very important, because usually 5 prime to 3 prime, 3 prime to 5 prime bonding is seen, but here 5 prime to 5 prime to 5 prime triphosphate bonding is seen. Again I am trying to explain the method. This is the mRNA and this is the N terminal, and it has 3 phosphate, alpha, beta and gamma. Okay, 5 prime triphosphate group. then what will happen? One phosphate group will be removed from the gamma region. Okay. Next what will happen? Only alpha and beta will be remaining. Next, guanyl transferase, that is an enzyme which transfers the phosphate from the GTP. GTP full form is guanyl triphosphate. So in the guanyl triphosphate, beta and gamma will be removed and the alpha phosphate will be attached to it. After that, the guanine 7-methyl transferase will take place by the help of adomate, which is an enzyme, and it will donate a methyl, and thus at the 7th position the methyl group will be attached. This is known as the C0 capping, which is very important. Okay, it is asked many times. Next, 2' methyl transferase will take place, and here the 7 at the 2nd position the methyl group will be attached, and this is known as cap 1 position. Okay, and the bond which is formed is known as 5' to 5' triphosphate bond. This is all about the capping process. Then what is the function of it? It protects the mRNA from the degradation, because as soon as the mRNA will be emerging, you can see here, as soon as the mRNA will be emerging, many nuclease enzyme will be present in the surrounding. It will attack and degrade the RNA. So there will be no use of it. So to protect from the attack of the 5' to 3' exonuclease activity, it will help the mRNA protection, as well as it will also recruit protein for the mRNA splicing and polyadenylation, which is very important process for modification as well. And capping helps us to distinguish between the others mRNA, because there are different mRNA, that is rRNA, tRNA are also present. So the capping process helps us to distinguish between the mRNA and the other RNAs which are present in the nucleus or the cell, you can say. So this is all about the first modification process, that is capping. Moving to the second modification process, that is polyadenylation. What is polyadenylation? That is addition of poly A tail to the 3' end of the mRNA sequence. It is a long chain of adenine. Adenine is a nitrogen base which is added to the mRNA molecule during the RNA processing. It occurs, when does it occur? This is very important. Before mRNA leaves from the nucleus, because once mRNA leaves to nucleus, it will reach to the cytoplasm, and then the process of protein synthesis will take place. Before that, the mature mRNA is needed to be performed, so that before mRNA leaves the nucleus, the poly A tail will be added. The position of the 3' end of each mRNA molecule is specified by the signal. So the 3' end has the signal which is present. You can see here in this, there is a signal will be present. Cleavage and poly A signal will be present at the mRNA sequence. When the RNA polymerase moves along the sequence, when it will reach this sequence, then polyadenylation will take place. We will be coming to this part. Let us first understand the theory part first. These signals are transcribed into RNA by RNA polymerase II and a series of RBP, that is RBP and RNA processing enzymes bind to it. Okay, you can see here in this diagram also that the RNA, once it is moving and elongation is taking and its transcription is taking place, once it comes to this signal process, this signal where the poly A signals are present, there will be binding of protein like CPSF, CTSF will be binded to the this sequence, and the polyadenylation process is taking place. What are all they? We will be discussing each. So there are two proteins which are responsible in the process of polyadenylation. First is the CSTF. What is the function of it? It will cleavage and stimulation factor. CSTF full form is cleavage and stimulation factor, and CPSF is cleavage and polyadenylation specificity factor. It binds to the AAUU sequence and has hydrolase activity. What will we be doing? Let's see. Addition is done by the poly A polymerase, that is PAP. So the PAP protein or polymerase will help in the addition of the adenine sequence, and it is a template free. Okay, it does not depend on template and also does not act on the extreme end of the 3' end. So remember that the 3' end polyadenylation will take place, but not on the extreme end. Apart from that, there are other protein also present like the PAB, which is known as poly A polymerase binding protein, that stimulate the activity of polymerase A. That is, polymerase A activity will be helping in the extra effort. Okay. XRN2, that is the exoribase nucleus, which degrade the uncapped mRNA. So after the mRNA is finally done, the portion of the mRNA which is uncapped will be degraded by this XRN2. Okay. Then PAP adds one at a time 200 aminonucleotides at the 3' end and proceeded by the cleavage with the help of ATP and AMP, and poly A binding protein determine the length of the tail. Okay, let's see the process. So first what happens, this is a sequence, this is very important, AAUUAA sequence. Okay, and this is 10 to 30 nucleotide base sequence, and this is a GU rich sequence which is known as consistent sequence, which will direct the cleavage process. Okay, so what happens, certain cleavage factors CF2, CF1 as well as the CSFP, that is this two, that is CSTF and CPSF will also bind to it, and the cleavage will take place here at this end, and the polyadenylation tail will be added, up to 200 nucleotide will be added to it. Okay, so see here, this is the RNA polymerase, this is the tail region, here already the CPSF and the CSTF will be present. When this RNA polymerase will reach this signal end, then what will happen, these factors will be attached to it and the polyadenylation will be beginning. Okay, you can see here the CPSF, I have already told you, will bind to the AAUAA sequence, and then what will happen, the PAP protein will add the adenine nucleotide as well as determine the PAP protein will add this adenine nucleotide, addition of poly A binding protein, then the poly A polymerase will regulate the length. Okay, how much length of the polymerase will be formed, it will regulate, and the mature mRNA will be seen like this. Okay, again I am trying to explain what will happen. There will be a signal at the end of the 3' end. You can see here it is not present very end of the 3' end, slightly before the 3' end there will be a sequence which we have the poly A signals that encoded in the DNA, and there also the CPSF and CTSF, that is factors, are already present here in the CTD region. Okay, and once when this polymerase will bind to this sequence, immediately the polyadenylation process will begin, and the CPSF will bind to this sequence, and the PAP protein will bind and start adding the nucleotide, and also the poly A sequence, that is poly A binding, will determine the length of the final length. What will be the final length of the tail, that is 200 to 250 nucleotide will be determined by them. This is all about the polyadenylation step, and what is the function of it? It will make the RNA more stable and prevent from degradation. Both capping and vessel as well as polyadenylation has the main process is to determine the stability of the mRNA, so that it will be helping us to make the protein. Okay, this is the main process. It will prevent degradation and allow the mature mRNA to be transported from nucleus into the ribosome, where the actual process of protein synthesis will begin, as well as it will recognize signal for binding to the translation factors. So the poly A sequence had certain signal sequence which helps for the translation process also. You can, by this you can determine how much the capping and the polyadenylation process is very important for the protein synthesis. So that's much for today, and if you have not watched the process of transcription, I do recommend to first watch those two parts, that is transcription in prokaryote, eukaryote, then come to this processing part for the better understanding. In the next part of the video, we will be discussing about the processes, that is the splicing process, and then the post transcriptional process will be coming to end. And that's all to today. Thank you, and do subscribe, like and share as much as you can among your friends. Thank you and have a nice day.